rabies by the French biologist Louis Pasteur. So it's an anniversary of the first successful ever vaccine for zoonotic disease. This Louis Pasteur's work not only saved a number of humans, but also initiated an effort to understand and prevent the zoonotic diseases. According to the FAO and CDC, 60% of all human infectious diseases are of zoonotic origins. Therefore, promoting awareness and designing a framework for the preparedness and responses to the zoonotic infections is very important. So today we have our distinguished guest, Dr. N.H. Mohan, Director, NBAGR, Colonel, to give more insight into this topic. So first I request Dr. G. Taru Sharma, Director in IIB, to formally welcome Dr. Mohan.
Thank you very much, Dr. Abhijit. Good morning to everybody. Well, I think I'm really happy Dr. Abhijit has introduced the importance of this day. And as he said that tomorrow we have Dr. Lalji Singh Memorial Lecture. And also we have a spotlight on 5th. So there are a battery of important events, I would just say, but let me get focused to this. Usually this institute has been inviting a veterinarian and a medico together, or the scientists who are linked with medical sciences research and the veterinary sciences research. And this is how we have been just trying to understand and celebrate the importance of this day. This day is, as has been told by Dr. Abhijit, I think we move ahead with that. But my task right now is to greet everybody and formally welcome Dr. N.H. Mohan, the Director of National Institute for National Animal Genetic Resource. It's a bureau, National Bureau for Animal Genetic Resources. So there are two bureaus, one for plant genetic resources and another one is a bureau for animal genetic resources. So Dr. Mohan used to work a lot on a climate change, and before coming over here, he was working at NRC PIG. And some bigger projects, he was trying to corroborate the climate change data with the production and production linked traits. And I'm sure that this day and his data and his talk would be good to all of us, and we look forward. Thank you so much, Dr. Mohan, to make it for the day and to join us. I mean, I'm also happy that there were meetings at Telangana Veterinary University and Poultry Institute, so it could be clubbed. And many thanks. We look forward. Thank you very much.
Thank you, ma'am. Just a brief introduction about Dr. Mohan. Dr. Mohan is currently Director at NABGR, Colonel. Dr. Mohan is a trained veterinarian, did his graduation from Kerala Agriculture University in 1998, post-graduation from IVRI Izzatnagar in 2000, and PhD from Assam Agriculture University 2008. So after his PhD, he went for a postdoc at Massachusetts General Hospitals, Harvard Medical Schools, and also at the Institute of Molecular Biology, Mainz, Germany. Before becoming a director at NABGR, he was a ICR National Fellow and the Principal Scientist at National Research Center on Peak, Guwahati. His research interest lies in physiogenomics of stress in livestock, nutrigenomics, climate resistance agriculture, and sustainable management of animal genetic resources. So he has published more than 50 papers in international peer-reviewed journals and has 2 patents granted. He is a recipient of several awards. To name a few, Dr. P.K. Barkanath Memorial Young Scientist Award, Young Investigator Award from the Indian Society of Nutrigenetics and Nutrigenomics, ICR Lal Bahadur Shastri Outstanding Young Scientist Award, ICR Farukuddin Ali Ahmed Award for Outstanding Research in Tribal Farming Systems, and Professor M.N. Razdan Memorial Mid-Career Award from Society of Animal Physiologists of India. He is also a member of several national and international societies. So with this brief introduction, I would like to invite our distinguished guest, Dr. Mohan, to deliver his talk on climate change. So I think this is a different one. Okay, and zoonotic preparedness. Yes, Dr. Mohan, please.
Yes. प्रेजेंटेशन नहीं आएगा क्या? ये क्या करना है? माइक इधर रख दीजिए। हाँ ठीक है, उसको भी करना है, ठीक है। देखना थोड़ा मुश्किल है। आप उधर जाके प्रेजेंट कर दीजिए। हाँ, that I can. That I can. Good morning, respected Director NAB and esteemed scientists and other faculty members of the institution. Thank you very much for inviting me for a lecture and this providing the platform to have a interaction. And rather, I am very much privileged, I feel, because ma'am used to be my teacher. We have been opportunity to work together also for a period of like 2-3 years. And we have a very long, 30 years, almost 3-decade old relationship, professional relationship we had. So that's a really a privileged moment for me to present in front of your own teachers or your own colleagues. That's a great thing. Thank you, ma'am, and thank you, NAB. And there are small 3 presentations have been just made. One is the climate change and zoonosis, and second is a little summary of my background work and all ma'am was discussing so that people get aware of those areas. And the third is little about my present institution, National Bureau of Animal Genetic Resources. So coming to the topic per se, it is really a customary thing that we start with what we have as a livestock wealth, because it is just to create awareness. And we have a very sizable livestock population in the country, and this is a true wealth, actually, true wealth. And this institution also a part of a custodian of really say the genetic resources. So this becomes a, as you can see that even though this is 850 million poultry and this 536 million animals, these are, say, this is a big responsibility. These animals we have to maintain, we have to, these are actually the animals which will provide animal source food for the billions of population. We have a long projections. As you can see that the share of the livestock to the total agriculture is like 31%. It is actually progressively increasing. That means that it is the driver of the driver engine of the basically the agricultural production. None of the plays a very key role in coming days, because in 2050, of course, it's a very small thing, but how that population will be, what will be the future projection? It is said that this will be more than 1.65 now. A latest figure I saw 1.74 I have seen, but will be our population. Most of them will be living in the urban areas, and there is also a income increase. So food pattern will change. So these are the projections of the Niti Aayog, where the global share of agriculture export is there. But what we are endowed with is only 2.4% of the land area and only 4% of the water resources. So we have our own limitations, but we have our own strength. But this also, we should not forget that this hunger index, food wastage, these are some of the very worrying concerns for all of us. And of course, the gross contribution is 31%. I have already came. And the share of the AS of agriculture, this animal source food will become double, actually. Now it is 8%, now it will become 16%. So we have, and we also have a lot of opportunities coming in all those areas in value addition. These are opportunities actually for this institute also to look into. So livestock sector is really poised for, there is a huge demand of animal source food will come. This will come from this 536. Maybe we will have a little bit more coming. We recently had prepared a document on 2020-50 livestock sector for it's a policy document. So we are expecting like it will be like 800 something animals will be there, even though the growth of cattle and other thing will be gradually happening. So the demand of dairy products and our share of global share is like milk, it is 24 and it is projected to be like 30. So, and very robust feed, uh, animal feed market is there, which is poised to become, it's in billions. So we really do not realize the potential. There are a lot of scope actually in everything. Okay, coming to the endangered livestock, we are 229. That is our institute's main job. 229 livestock breeds have been registered. I will come to that. So this is something like all India food availability is like out of where we have milk, we are good, but meat and egg we are still lagging behind the ICMR recommendations. These are some of the things. If you really see where the we have to work on is the milk. It is now 230, 240 MT is there. From there, it has to go to 550. And meat from 9.7, it has to go to 18.7. These are the projections of Niti Aayog. These are the challenges which are ahead. Along with that, this and all thing comes. Coming to the topic per se, global warming, and I will not go deep into all those because everybody is very well aware, and those persons who do not believe that it was never happening, they also now believe. I will start with my personal own experience. I will start with when I went first to Guwahati. It was March month, 2003. So we had, I went from Ayodhya, Faizabad. So it was, March was very cold month. Usually March should not have been cold because that time it used to be cold in Guwahati. Then subsequently up to April, May, that year we used Rajai or thick blankets we were using because it was cold. And that time Upper Shillong and all used to have snowfall. Believe me, past 5 years, I never used blankets or thick rajai or anything in Guwahati. It was so comfortable. It is not required because the climate has really changed. Urbanization is there, that also have contributed, but certainly the climate change, what is real, that is also happening. This is my own experience of this 20-25 years stayed there. So really we can say. And the worrying thing is that how much it will be hardly hitting our GDP. This ADB has calculated that 25% of the GDP will actually go for only mitigating this kind of. So we have a lot of 1.5 degrees. It is like a standard. It has been told that in coming days that much change will come and that will cause a lot of irreversible changes. This is a beautiful article from Nature which shows how you all know that I'm not going too deep into it. So a lot of number of the events, adverse events like high precipitation, drought, these all will increase actually. That we all very well clearly know. And it is found that more than 61% of the last year had heavy precipitation than the what is expected from normally. And this is the paradox. 48% had a drought actually. 48% of the land masses in the all over the globe. So it is like both extremes are there, which actually drought is leading to heat waves, fire. So these all have not only impact the human lives or animal lives, this also can cause other type human mortality. Oh, that is a really worrying thing. More than 65, it is estimated that 65%, those who are above 65, there is a death rate of like almost double, 1.67 times from the highest, just because of these heat related causes. So this is really a very matter of great worry. Ask this in 1.1.5, this is an indicator of this disease. So it's a Lancet. One should be able to see that it's a beautiful Lancet series. Every year they used to release very good document. So these are real worries where in this background we see that how many deaths, how much crop area, and how much damage can happen over a span of time. And this is really a matter of worrying thing. I'm not going to just, I would say one sentence says, this is from Lancet, one sentence says that it's food security will be affected, and it will be access to sanitation, water, all will be going to be affected, and sea surface, reduce oxygenation, and we know that these all things are being expected. And In fact, those who have and who have not, that difference will continue to increase because the accessibility is a main factor in when this kind of issues come up. So, subsistence farmers and indigenous people for whom food availability will be so sensitive. So these are, so along with that, animal production, of course, I'm not going to, this itself is a separate topic of a presentation, direct loss of productivity and the indirect loss through the feed availability, feed accessibility, quality, and disease distribution. So coming to the real, main topic of this mainstream topic, zoonotic diseases and climate change. Zoonosis, we all know that these are diseases commonly shared between animals and humans. And the very interesting thing about this is now the emergence of those diseases which were not really a human disease or an animal disease can cause spillover. There is a spillover is there. So what are the reasons? There are so many reasons like ecosystem changes, habit loss, global warming, deforestation, and along with that, the biological characteristics of the pathogens, vectors, so they are also changing. So both are actually really acting together to cause this havoc. As we know that even though it is said that past 650,000 years, we had like 7 galactic cycles of thaw and frozen, and we had like some 15,000, some or 11,700 years back that ended. Then we humans and other things that survivable, but the relics of past can really come and haunt us. That is, we will see in some of these slides because some frozen areas are still getting thawed, and that is posing some very worrying questions to us. And zoonotic disease, as you can see that in the beginning itself, somebody told like 60, so 1% of the infections are zoonotic. Now, in a latest one, I saw that 75% of the emerging diseases, I also put that original figure what you told. 75% of the emerging infectious diseases are zoonotic. So, along with the alteration in the host, vector, pathogen, these dynamics, and of course compounded with wildlife, we are the deforestation, the animals will come into the animal human interaction is also further increased. So this all adds to the cause of these spillovers. And this is just a model about the dynamics of how the host interacts with a not endemic human population and the effective population, and how we closely interact with the host niche. So infected host, whenever that interacts with the human population, this results in the actually infection, and that infection can persist for a very long time. And the globalization and movement of person from one area to another, so all those actually contribute to the spread of the diseases. Also, there are several socioeconomic drivers are there which also cause that thing to further propagate. And this is based on a study in Ebola, this diagram. So this is a general picture, but it's a very beautiful article in ISME Communications, because this COVID-19 have made us to rethink so much on the, so there is a proposal, there is something like a hypothesis from bat it came, and all those, there are many hypotheses about that. We don't go into that, but that disease has really made it to think that there can be a close association, much more than what we, because wildlife resource reservoirs are there, they actually have got a much more important role than what we might think to. And we have a lot of peri-urban species are there, so this close interaction, this also gives a lot of opportunities for the wildlife spillover and the continuation of the sylvitic and peri-urban cycles of these diseases. And there will be secondary reservoirs of these various pests or the pathogens coming. So there is a chance of the it is further increasing, because this is a background which even if climate change is not there, it will be there. But how the climate change matters is really every actually, as you are aware that the zoonosis, it can be a waterborne disease, or it can be a vector-borne or airborne, foodborne. There are many modes of transmissions are there. These are some of the examples, because there will be, it can be a course, actually 6 months course, that zoonosis and climate change. So I will got some specific examples, that dengue. Dengue is one of the, it's a true sense, it is an emerging zoonotic now, it is considered. And this Aedes, the Aedes, I think you are all aware that these mosquitoes are vectors for more than 50 diseases. So lot of diseases are there where mosquito can act as a very good source actually. So it is observed that the chances of, because of the only dynamics of this climate change, these animals, these Aedes, albopictus and aegypti, that the increase in the disease by 46 and 10.7% between this 1950 and 23. This is being attributed to the characteristics of the vector, not the other one. Similar trend is for other diseases like Zika or Plasmodium, chikungunya, many diseases actually, West Nile. So there are many dimensions for that, and Ebola and all. Ebola, this actually there is a spread actually. So there is a slide on that the spill to the areas where previously those infections were not there. There is a example, it's from NRCP, my previous institute, Japanese encephalitis in Assam. This is a classical study. It was observed that JE is very much related to Assam. A lot of flooding is there. Then we had a paddy cultivation. So these are close related to the mosquito population. So mosquito population is related to JE incidence. So a classical textbook-like study we had, and we were very amused to see that. See, this is reality. It's not like a first-hand data. So really, the JE typically is a disease where mosquito plays a very key role. So these are some of the examples where we can see that many diseases, many things have been actually related to various parameters like West Nile disease or tick-borne encephalitis, many diseases. So these have been related with several. These are just to show that there are multiple parameters. The meteorological parameters like say sunshine or temperature or rainfall or humidity. So if you take, each one has got a separate actually relation with each of the diseases. So they affect them in a different manner. So but overall, if you see that temperature, precipitation, humidity plays a very key role in the distribution of, we will come to that. This is just a study where it's in mid-archive. It is in recently they have screened several, it's like a meta-analysis. They did a large-scale study of these observations, then summarized. See, as we can see that the Southeast Asian region is also very vulnerable particularly for the temperature, precipitation, humidity, and incidences of various diseases as compared to the rest of the thing. So that is a little worry thing, which all our neighborhood. So this is a similar type of study on same author study, where he also says that these are the hantavirus and arbovirus. So many things which the incidence of which were very less are, we should actually anticipate that this all come, and there is a chance that there is a high probability that the trend goes. And of course, when we go to specific cases, if I say that formerly, the former regions where it was cold regions, or some regions where the temperature was not very congenial for that organisms to multiply or grow, now they have become hospital, that being good places for the organism to proliferate, and they are affecting. This is Crimean Congo hemorrhagic fever, Rift Valley fever. These are classical examples recently observed that they are appearing in those areas which were not there. So this is one of the effect of the climate change in relation with the zoonotic, affecting the other areas which were not typically affected. And second is widening of the effect. So we thought that, okay, we might be remembering that before monsoon, some animals or something will be injected for HS vaccine or something, so that we prevent that particular before that. So you take a flu shot, it might be, so before that flu comes. So here, that period when we can get, say, monsoon, not monsoon, now you can get at any period of the year. So that widening of the time frame is the second thing. Third one is the opportunities of spillover. So there is a very high chance that we get infected when we probably travel to one place. So these are some of the things, classical examples. There's Lyme disease, dengue, Nipah. Nipah is one thing very really we have to be very cautious because pig has got a very important role here. And we have, I think, few cases in Kerala and southern region about the Nipah. And the similar cases for, I am just saving one example in vector-borne diseases. Again, vectors are really, climate change has got a very, very intimate relation with the vectors because arthropods being ectothermic, the temperature influences their behavior, their activity, their reproduction. So further, we can expect that they, of course, heavy rainfall has got a different effect. So that gives a vegetation cover. So that gives a good chance for more mosquitoes or those kind of things, water logging. So this all gives further breeding ground, fresh ground for the mosquito-borne zoonosis or all those things, because Aedes species, it alone is, it is reported that more than 22 arboviruses. So it's a lot of, it's a very, and just see, by 2085, it is expected that 50 to 60% of the population will be at the risk of dengue population compared to 35%. So it's a projection, long-term projection from now 25 to 85. So 60 years projection says that the vulnerability will be so high. Like from 35, it will go tick-borne, same case as the other vector-borne. So same, the vector characteristics, the insect characteristics will be affected by the climate, and in turn that will affect the zoonosis and waterborne zoonosis. This is also a very big worry because those areas were previously not flooded because of heavy precipitation that are getting flooded. And of course, you all understand that the flooding means there is a very high risk of contamination with the sewerage and other water bodies. So this in turn has got a very high possibility of increasing the emerging and re-emerging waterborne zoonotic diseases, especially this Campylobacter E. coli O157, then Cryptosporidium giardia, and it's a very long list actually. This is a very, so the difficult thing is that the list of the disease becomes so exhaustive, and the monitoring also have to be such robust that for all we should be looking in coming days. Foodborne, it is actually concurrently with the waterborne. Foodborne is also becomes and a typical thing is Campylobacter and Salmonellosis. So these are some of the real worrying situations where the temperatures, precipitation, and all meteorological parameters, inconsistencies of them or the variability of these parameters influence the diseases. Rodent is also almost similar to the vectors, where there are several diseases that El Nino associated northern oscillation has, it is found that it plays a very key role with this anomaly has got to play with the different rodent-borne diseases. So rodents, again, the habitat, see, rodent survival, rodent human, we also have got a very important role. Anthropogenic activities also influence these vectors as well as rodents. So again, it says that one of the reports says that 1% increase in temperature could increase in 50% of the Yersinia pestis. That is you understand Yersinia pestis, and it is over. So it's a like a classical example of what we have read in book. Now it's actually, we can see that in any disease or most of the diseases you take, that can happen. Airborne zoonosis, needless to say that wind really plays a major role. And one of the thing is this highly pathogenic avian influenza virus, tuberculosis, and this distribution and the movement of air also plays a very key role. And needless to say that this causes the organism to move over large areas to infect those populations which are vulnerable, or which probably you can understand that tuberculosis is very intimately related with our dietary pattern and the quality of the nutrition, what do you have, and the immunity of the body. So those who are vulnerable to those, or they have previously, they have immune deficiency disorder, they become more vulnerable because earlier. So there is a very high, that's why this term earlier I have named it in my title in a different way. Then I thought that this is a very beautiful way. Somewhere I read actually climate change is a real super driver. Lot of okay. One of the thing, I'm not going into deep on that. This is a lot of opportunity for this institution also to develop a lot of these models for the different zoonotic disease prediction models, because for each disease there may be a different set of meteorological parameters. Apart from that common they may be there, but the intensity of the influence of each parameter will vary. So this is an opportunity for this institution also to take care of this development of models would be have a very it will be a very useful contribution. It will be a great contribution, rather, I would say, because this is the one which will help in the health policy planning. So this is one paper I saw, how the pathogen, how it is modified. It's a classical example is this human this influenza virus, because genetic reassortment is possible for this, and there are emergence of different H and N's combinations and emergence of different diseases, and they have got a profound the entire, especially it is true for the one which are with the RNA as the genetic material. So RNA viruses have got much more very high chance of getting altered, and yes, these are some of the great worries and a lot of opportunities here again to study or detect, because this also helps in the phylogeny and you can trace back the infection. So you trace back from where it has emerged and how it has emerged. So it can really give a lot of, this is a very beautiful reference, climate change and influenza, very nice paper I have gone through while preparing these slides. It's a scooping review, very nice review. So these are some of the real concerns. So this is a pathogenic, it's a from nature. This you can see that there are several, this is again a meta-analysis, several parameters they analyzed, and these lines, the thickness of the lines directly are related to the diseases. So these are a large number of diseases are there which are actually influenced by the influence of each meteorological parameter is different on each disease. So it becomes an opportunity to develop models for each one of them, other than having a common model. Common model also is an important one broadly, but each one has got a specific relation. It's a very beautiful. It's a not only zoonotic, but it covers other diseases as well. It's a general disease. So as you know that this doodle says the larger size or smaller size indicates the general relationship of the each parameter. On the right hand, left hand side, we can see warming, precipitation, floods, different parameters are there, how they influence the various diseases. The extent of influence is different for each disease, and the meteorological parameter, even the duration of the precipitation, duration of droughts, that is the spell, these all have got a profound influence on the development of zoonosis. So it is said that 58% of the infectious diseases confronted, this is the exact wording from paper. I just thought really they say that it will continue to be influenced by these parameters, and 16% actually diminished. So that can also happen. So both ways it is there, but 16 and 58 are no comparison. It means much more, they are higher than this. And they identified that more than 1,000 pathways are there, climatic hazards, where the pathogen can have a profound influence on the disease incidence. It is from this Nature paper. Okay. Having said this is a much more worrying condition. Much more worrying condition means it's a science on full volume is there on this, lurking in deep freeze. It's a title page of the cover page of the Science magazine. It says that the areas which are previously frozen, permafrost areas, bacteria, viruses, these pathogens have been there previously frozen. Because of the melting of those ice areas, these pathogens are coming out. They have identified different pathogens which were not reported. See, this is a very, here we have in Himalayas, there is a possibility. There are people, I couldn't find a report from Himalayas, but there are other reports. This Arctic permafrost is a classical example. There is a full article on that. This is a National Academy Press have got a full book on that, understanding and responding to global health security risks from microbial threat in the Arctic. Most of them, this is a very good study can happen if you can trace this glaciers or all those areas in Himalaya. So there is a lot of opportunity, because we really do not know. There is a one European consortium, this versatile emerging disease observatory, is that they are studying the northern latitude and warning for it's a classical example. And this one of they say that in 2016 Bacillus anthracis was there buried, And suddenly it came in Siberia, and so many animals died. And there is one story here. It is in 1773. There was coronation of Denmark king. So one girl and one boy were sent to attend that from Greenland. They stayed there for some time. In September, they came back. So en route, that girl died because of some disease. 1773. So it is reported by, it's in, documented by a missionary who traveled, and the book is available. After some years, after some time, that boy also died. Boy had some cutaneous lesions, because that time it was not known what it is. So now it is traced that it was smallpox. It was smallpox, and in by summer, by June, the entire lot of population died of smallpox, because this boy has brought that they were really virgin, and the pathogen fully took over, and there were dead bodies lying. This is a documented by the missionary. After seeing this, see, these bodies, many of them might be still be frozen and still in the behind ice, because it's not. There's a very high possibility of not only this, any disease, pathogen, bacteria, and all those things could be. See, it's a very interesting study. Then they have identified from a particular frozen region. I have just taken from that, and these are like very new viruses. They have been identified from this permafrost in the Siberia. Very interesting paper, Olympic et al. In 2023 paper. They have done a electron microscopic picture of that. Very viruses, Pandora virus, Cetra virus, Mega virus. Of course, I have not heard about any of those. I have not studied during any of the courses. So these are like new viruses which have been updated. That paper itself says that an update on the eukaryotic viruses derived from permafrost. Very interesting study. So we have a lot of opportunities here to study all of them. And there is another one, this Beta Geica crater, you might be knowing, is known as Earth's gateway to hell. It's growing at 35 million cubic feet per year. It was a line like in 1962. Now you see a very big crater. This crater is really very huge. Here we have other worries also, other than the bacteria, viruses, and these pathogens, or the so-called microorganisms inside. Has this getting defrosted, what happens? The adjoining areas fall into that. The crater enlarges. Plus, it poses a very unique situation. The organic matter, which is already there, decomposed on exposure, it actually generates a lot of methane, carbon dioxide. This also contributes to the global warming further. So there are multiple worries actually, if you really say. So this is I leave it for the thought of the house, that how really these are real worries and how we are going to take this. Is because we have to understand that we are at a tipping point where the life is at a very critical stage, because this may be the 1,000 years or 5,000 years may not be very big in the case of a history of a Earth or say planet. This is nothing actually. We have been very recently we have come to this place. So it is not a very long time that if we are not properly conducting ours that way, we can become extinct. Not a doubt. One virus should be sufficient. We do not know actually. So these are some of so what we might require is, along with the climate change, those kind of precautions, other long-term policy decisions should be there for identifying, monitoring, warning, all those things. But see, whatsoever we might say, it's a fact. Again, a recent report today morning only, I was just reading that 2000 last year was the year where the highest fossil emission was there. 2023 was the highest. So we have been talking for many years, greenhouse gas emission. Fossil fuels were maximally used during that, just a year back. So that means the speech and the actions are not really in sync. So we need some more proactive steps from our end to take care of that. So such institutions like your institutions that can really help in that policy development and those modeling, which can really help in the disease surveillance at a short term and a long term. And we all need actually a proactive approach, awareness. See, one of the classical diseases where we could never eliminate was rabies. Rabies continues to really take away many people actually. Many people, dog bites are quite common. Dog bite infection, correct? We have very robust vaccination for that. Why? So these are some of the, I leave it for the wisdom of the house, that these are some of the worries where the development of models, because each one of them can be a very detailed topic of discussion. So I don't go into those criticalities of that, but I leave it to the wisdom of the house and the scientists that we collectively act together to not only mitigate this climate change, and those climate change actions also concurrently should address health concerns. So this is like a one beautiful opportunity will be to collaborate across the globe, different organizations, different opportunities. I do not know where our animal health thing has ever explored the Himalayan and those frozen areas for in search of these organisms. Could be a great opportunity. Not only a science paper waiting, but there could be a good opportunity of information coming out. But you should be cautious. These are unexplored organisms. Knowing anything about them can also open a Pandora's box. Thank you. I stop this thing here. Then, if anything, the interaction means we can have now, or I wanted to, as I will finish off. There is a small presentations about one of my work areas, because I am not a great like a zoonotic guy. दूसरा slide डालेंगे, एक summary है. This I will not this I will not go into detail of the research. I just wanted to give you an overall view of what I have been working. Just to give you an idea. Just to not much results I will go into, but this is just to so that we can work on at a future date. I have been this I will not go. I've been working on, these are some of the works, important works immediately I undertook in past some years, say like 10 years or so. One was the whole genome analysis of the different breeds of pigs. Then we also developed methylome of pigs, high quality genome sequencing we did. We analyzed them. Then we also screened a lot of animals for the identification of relatively heat tolerance traits, and we developed an array for screening those animals. Then a lot of proteome metabolome studies we undertook. We also analyzed the microRNA, spermatozoa, like these all areas. By and large, most of the time was related to the heat shock and climate change. That was the key areas. So there are only 2-3 slides about the genome. I say we did the variability. See, there are one important thing I will just like to say. Lot of heat shock variant protein variants we have identified in Gubru and Mali, which are indigenous animals, as compared to the Hampshire and large weight Yorkshire. These are exotic animals. So there are a lot of unique characteristics, unique variants are there, which could be explored for as development of heat resistance or these animals. This was one important finding of that. And we identified a very hyper variability region in the chromosome number X, 10th X chromosome. We have identified in between 43 and 86 MB region. As you can see that the variability is very high. That in exotic animals, we don't see that kind of a variability coming, because it's very clear actually. There is a this is a very important region which we can be explored for identifying those genes could play a very important role, because this is only present in the Southeast Asian animals. This is also seen in mission. So this is a very important find. These were the very important findings of the study. This is the hypervariability region, because you really do not see in exotic animals. So this is one thing. A lot of genes are there. There were 300 plus genes in that area. Then we also compared the pan-genome comparison with the different species that we undertook. Then we also identified a fertility region in chromosome number 7 located between 10 to 11 MB. It's a 0.34 MB region. This was one of the findings of that study. So that is about only that. Then other study, we identified a heat shock responsive region in the DNA because we did the chip of where the heat shock factor binds. Then we, based on that chip, we identified a 21 nucleotide sequence in the genome where the HSF can bind and elicit the heat shock response. And of course, you can see that this is associated with not like only heat shock factor, that HSF is not only related with our HSPs. There are so many genes, 1643 genes are there with whom that particular heat shock factor is associated. So that means the whole machinery, a heat shock or the thermal shock can actually alter a lot of genes. Lot of genes can be affected because the promoter, it binds in the promoters of 1643 genes, because that second one was that there is a serine 121 methylation, that not methylation, phosphorylation also was one of the controlling areas where the HSF activity could be regulated. So this was one of the, I'm just quickly I'm showing another study. We compared the already adapted species like pig and goat of two different states from extremes, actually, Rajasthan and Assam. So we also found a very interesting picture about the uniqueness of the each species, how in long term, how they got adapted. We studied actually two species. So these were some of the studies we really cherished, because each one had got their own HSF1, HSF4. So those were in pigs, but other time the other one had HSF2 actually. So each one has got a unique way of regulating the adaptive response. In species-wise variation is there. Animal-wise also variation definitely is there, because variants are there. We have identified unique variants in the Indian breeds. So this is the array we developed for screening of the animals. Then we also identified some areas where the transcription factor, this transcription TSS is affected by the heat complexes, the ribosome quality control complex. See, there are a different way of controlling the regulation and the quality. So there are the heat shock. It's a, then we, one very interesting study we did, the microRNA distribution. We conducted one degradome analysis. So what we took, the one sentence I will say, the degradome studies, it gave a very important information into our this thing, because it is said that the mammalian or in animals, the miRNA destabilizes the microRNA, mRNA, and ultimately the regular chopping machinery comes into action and they just cleave it. So that means that miRNA is not directly actually cleaving it. But the degradome, when we did, we identified that endonucleocytic cleavage is also possible for some of the things, which is reported in plants but not in animals. So this is one of the first study where we identified that some of the miRNAs could also act in that way. It can cause instead of a direct destabilization, but cleavage can also be possible. And we predicted some structures that how that could destabilize. So some predictive studies, then proteome analysis we did. So a lot of studies, cell culture studies, and I'm not going into that, but just to give you an idea about. Then we also have got expertise on the development of the artificial proteins. We synthesized, we developed the proteins, especially this is a protein we developed for assaying the African swine fever, ASF disease. So we developed the epitopes we took, then we made a mala of that epitopes. Then epitopes we made it into a gene form. We put it into a vector. Then we express that thing. Then we are now using it for immunoassay. So this is one of the proteins where we artificially synthesize those proteins. So these were. Another one is the fertility thing. Very interesting study. We identified two transcriptome, large scale transcriptome and real time. We identified like 50 markers of fertility. Then we were very lucky that we had about 26 animals which were used for regular semen collection and insemination. Each of them were bred to hundreds of females. So we had a very unique opportunity to evaluate each of them. because all the animal, we say that, okay, they are very, since they are used for AI, they should be, all of them should be good. But all of them are really not good, because that is not resulting in the, it's not like a female factor. For that, we first developed a composite index, incorporating various parameters. Then we, markers, along with the markers, we correlated with, because it is a very difficult to relate a molecular data with the phenotypic data. So that was a very difficult thing. Then we developed these multi-dimensional plots to identify, and as a result, we identified that the mean litter size at birth, there are some genes which are having very high correlation. These are the things, there are positive markers. We identified positive markers of fertility as well as negative markers of fertility. So with that, we will be able to screen, or we can grade the animals based on this expression. That composite index was, the right side is the animal number. We had a lot of animals over a span. This study actually took about 6 years time. Because over a span of time, each of them were bred to several females. So litter size, all those data we had, we were lucky to get. Such a data we cannot get in humans. Impossible. So only in a systematic form where the generational interval is also less, we should be able to get such a data. So that is one of the studies. So this is a study on the muscle microtome. That was also a very interesting study where we explored the reasons for the less muscle development in the indigenous animals. Our study identified that the fusion, myoblast fusion, is restricted due to some reasons. We identified that promoter of that particular region is methylated. Because of that, MY5 and MYMK, this expression is not happening, or to that extent, which should be otherwise happening. So even though the number of the potential is very high with that indigenous animals, Mali, what we studied, but it didn't happen because of a possibility of, because of this probable reason that the promoter is methylated. So these were some of the very interesting studies. Then we also conducted some genome editing studies on role of VGFA. Then a lot of we did on ovarial this transcriptome dynamics. So that we have our expertise on the reproductive biology. We had good expertise on those, but you can see that then we also had a work on tick posture analysis on new scientists joined, and we took a, okay, another one. If you see the below one, there is a paper which came in the medical hypothesis, my paper. So can virophages be used for management of viral infection? So we thought, we gave a hypothesis that virophages are very small, so they could be, why can't it be used for tackling viral infection, especially those diseases where very high criticality is the high critical, like Ebola or maybe rabies or such diseases where the person is very terminally ill, the patient is terminally ill with no scope of other treatments can affect. So why can't such kind of thing? Because if a virophage is there, maybe it's a natural one or a artificial one, there is a possibility that can destroy that particular virus. And since the host is not there, then there is no role for the virophage. But since it cannot infect of course, the mammalian cell. So this is a hypothesis already. It's a published paper. So then we also did some economics, a lot of biophysics studies. So these were some of the areas where we worked on. And there is one work this way also, because I was very closely associated with the conservation. As a result, there is a species actually. I should say that there is a species in my name actually. So we have a hymenopteran species which is in that is Dicopomorpha mohani. So we have been working on different areas, and interests were very diverse. So this ultimately, this biodiversity interest made me to join NVAGR, should I say that way, because that was one of the key areas where climate change, these are the threatening areas for animal genetic resources. There is a short 4-5 slides in my presentation, because it's my moral and my duty to actually talk little about my institution. National Bureau of Animal Genetic Resources was originally developed in 1984. It's a little old institution, 40 years plus, but last 10, 12 years have been so significant in the registration document. This institution is the custodian of the livestock wealth. This is an institution which recognizes various breeds by name whatsoever you call. and this we have the responsibility of according the breed status. Finally, that leads to the notification. So this is the institution, because whatsoever, so that way for getting, it's a geographical, wherever Indian flag is there, we are there. That much is the, so we have been closely associated with our primary mission is that identification, characterization, and the documentation of the resources. And we are also associated very closely with the policy development. That is one of the mandate of our institution, where we develop policy for the management of ANGR, and we also license with the global organization FAO primarily, because we have got there are commitments from our institution. Indian commitments are there, which is executed by the or overseen by the NBAGR, and we are also the nodal points for, we are the monitoring points for the 2.5.1 and 2.5.2 SDGs are monitored by NBAGR. So, and we also have a national bovine genomic center, very huge facility, and we have a lot of wide expertise on all those areas. We are not large number. We are 36 posts are there, scientists, including the director and technical. We are now we are about 50% of the persons are in size. So we have this is the distribution of registered breeds. We have now 230 breeds registered, and we also do a lot of conservation in situ as well as ex situ conservation. We have facilities for the, we have a gene bank where we preserve the germplasm, and we also store in the form of somatic cells, or we store in the form of the semen. So we have like 3.7 lakh semen different from different breeds we have stored. So these are some of the activities. These are the breed notification. Once we recognize that as a breed, we accord a accession number. You can see it's a very long one. India Buffalo 000200 Mana 010. It's a very long number. That's the accession. It has a lot of parameters. So once that accession is granted, then Government of India does a notification based on the accession number. So basically, we have a little statutory responsibility to get it done. So these are the, just to show that these are the some of the, this is the register notification which came on 15th of April. So we have been very closely related with the this NBA, National Biodiversity Authority, for protection from the biopiracy. We also assist states in the different policy developments. There are several activities actually. It's a policy is like a 50% of our activities to provide policy support and monitoring. So we have the multiple activities actually. But this doesn't mean that we are lagging behind in our typical molecular biology or those works. Because we have three divisions. One is animal biotechnology division, there is animal genetics division, and the third division is animal genetic resource division. So they work in sync, because once you identify population, the biotechnology group and AG group can go for a phenotypic or molecular characterization, their properties, all those things. So, and we have developed a breed watch. This is one of the important assets for the country, because this has been used very widely to predict the breeds which are undergoing extinction, or the which are vulnerable for extinction, at risk. So there are various classifications. So 38 threatened breeds are there identified. 5 of them are very critical. So we give special emphasis because it's like a asset for the country. So there are multiple activities. So there is a zero non-descriptive mission is being implemented by my institute. And these are some of the, we have a network project about covering 33 centers. We coordinate that, we fund that centers. So director is the project coordinator of that. So we have a very big activities. We have been very closely associated with, for example, Karna sheep in JMK, like that depth of the country. We have not, Leh probably will be going to be the first state, first place where the zero non-descript. It will be probably, we will go for a announcement maybe in 1 hour time, because we have completely mapped those. So these are last year's little achievements, characterization. Every year actually we are characterizing, and it's like a slow process. We characterize completely, and last year was a pretty good year, 24-25. Lot of characterization happened, lot of registrations happened. We don't rush actually for this thing, but because it's a systematic process and it has to be done like a with a thought, well thought. So those kind of things we do. And thank you very much. I stop here. If anything is there, means you are always welcome. Yeah, please.
Nice overview of how the institute works, especially your institute, and associated other topics on climate change and its effect on zoonotic disease spread. So I'll ask one question related to the how this institution works. Especially, let's say you mentioned that the all animals livestock breed in Leh has been fully mapped. Okay. So what is the kind of protocol or steps that is taken to characterize all the livestock?
Okay.
Both phenotypic and genotypic characterization.
Yeah. Both.
How it is done?
Both.
Exactly.
I'll tell you. You mean the process of the registration? How we do that?
Like when you say characterization, means you are specifying a breed,
Yes.
and you're mentioning that there is
No, not breed. We are specifying a particular animal.
Mhm.
This is not a breed, and all will come later. If the population is sufficiently homogeneous, yes, and a sizable population is there with uniform characteristics, then we say it as a breed. Otherwise, it is a single animal or a group of animals. So group of animals, characterization can happen in two ways. One is a phenotypic characterization. Phenotypic characterization depends upon the each species of animal. We have breed descriptors are there. Breed descriptors will very detailed evaluation will be there for each animal. Even minutest variation can also cause a variation between the group of animals. This is not very. It can be a very even switch color of the tail, or maybe depression on the backside of the pole. The distance between inner canthus and the muscle. Even small thing can also cause a variation between the different breeds or different varieties. So everything actually matters.
This breed descriptors we used to study in animal genetics and breeding.
Yes.
So now this breed descriptors, how means what kind of positions they are? What means who are they?
Breed descriptors.
Correct.
These are breed descriptors. Even it starts with the length of the animal.
No, these are parameters.
These are parameters. Yes.
These are already specified.
No. Yes, we have developed that. We have developed based on the FAO, and there are standard ways to describe that breed descriptors. So based on that breed descriptors, they are evaluated.
And in Leh, all the livestock.
Yes. In Leh, yes, we have been actually doing many of the species. All India, it is like a 50% is almost covered.
And then there's like genome level.
No. Genome is a different thing. With genome level is a different thing. Genome is a altogether a different thing. First characterization is a phenotypic characterization. In plants, animals, fishes, and all, phenotypic characterization is considered still to be the gold standard. So, and earlier we had like FAO markers, 30 markers were there. You might be, if you have studied AGB, so you might be knowing them. So this first is phenotypic characterization. After that, based on phenotypic characterization, and those based on that, we fill in that breed descriptors. Length of the body, or length of the tail, color of the tail, color of the switch. I'm just these are examples, but it's a very long list. Based on those descriptors, we evaluate and we ask them that what is the sample size. Suppose yesterday we were Sinkara Godulu, there is a cattle here. So they were telling that there is about the 3 lakh population is there. So 10%, they have taken an number of like 2,219 animals that particular person has evaluated. Yesterday we were talking in the PV Narsarao Veterinary University. That much of animals he has evaluated, and out of these animals, some 3 lakh odd animals. So those were found to be homogeneous in characteristics. So now what he will do, he will fill the breed descriptors and he will fill the average of that 2,219 animals. After that is submitted, then we evaluate. We evaluate based on the application, then a physical team will go and evaluate those animals. Then the actual process starts. Then only the application is further considered for the pre-registration. There is a committee for that. That committee clears it. Then we give an accession number. Then we do further. Actually, we are not into the genotypic characterization for the identification at the as a germ plasm. First identification, that is not, of course, we will do it unless it is there is a dispute, there is something of if that uniqueness is not there, or we do not have sufficient that kind of a population size. Of course, in certain times, we may have very less number of animals, like 50 animals, 100 animals. There are breeds. It is like a livestock wealth of the countries for posterity or any reason we are supposed to maintain it. But now also the question of that is coming, whether all the animals or how many breeds we should really be conserving on a longer run.
And one more question, this you mentioned about germplasm storage for
Yes,
different breeds.
yes.
So nationally, is it storage in one place in that institute
No,
or in
no,
multiple
no, no,
places?
no, no. There are, okay, very good question. There is presently there is a central storage with us in NBAGR, and also many of the institutions which we are working with, they have their own little storage of specific germplasm. And I can tell you, there is a one full-fledged gene bank, another gene bank is coming up in our campus. Plus, there is one more national gene bank is being developed in some area. Now, that area has yet to be finalized, as declared by the Honorable Finance Minister during last budget. You might be knowing that will be a combined facility which is being built for animals, plants, and fishes. That will probably in an area which is very far from the present location, so that even if some loss is happening here, that should not be yes, it should be in multiple locations ideally, is it not?
Thanks, Dr. Mohan, for the nice talk.
Please.
So you showed there was some genetic variation between breeds at the X chromosome.
Yes.
Right. Did you also do for the methylome of different breeds?
We have done, yes. We have made a long.
So my question is, let's say you should take two breeds, let's say indigenous breeds, not exotic and indigenous, but two indigenous breeds. Is the genetic variation more or epigenetic variation more?
Oh, very interesting question. Genetic variation is more. Yeah,
I mean, it's difficult to quantify.
It is very difficult to quantify.
Normalize.
Very, yeah. That when the methyl, that's why I didn't show the results of methylome. Actually, when that analysis was happening, I moved to that other institute. But we have very interesting results coming up from methylome, because that was a single nucleotide, but that was a deep sequencing. We did like 60x of single nucleotide bisulfite sequencing. So when you overlay with that regular genome, that gives a very interesting story. And that gives a lot of insights into the regulation of the genome and the variants.
Is there a paper published on that or not?
No. Paper is just in the process. It is with the NCBI. We thought once NCBI curates, NCBI has curated now. So almost in the final stages. So once after that, we will just immediately communicate, because then it will be that genome will be publicly available to everybody.
Thank you.
Thank you, sir. Thank you.
Excuse me, sir. Here, sir. So excellent talk, sir. So my question is this, that is your group is still working on developmental disorders in pig?
No,
in particularly this TET family, this 10, 11 elongation factors.
No, sir. No. Wow.
Why I ask this, because we are having a paper.
Okay.
And it's all in silico study, because we were actually not able to find the samples.
Okay.
So we identified a couple of key molecular pairs which in silico for computational.
Okay.
But we wish to validate them.
That might be definitely
But
be
we
possible.
wrote several times to NRC PEG, but
you were what kind of samples you were anticipating?
Basically embryo samples for 1 week, 2 week, 3 week.
Okay.
So these type of samples, if
Embryo means you wanted to take out from the live animals,
if it is
or
possible,
aborted ones. about
that
the
transcriptome was actually we analyzed the publicly available transcriptome, and that transcriptome was of exotic weeds aborted.
Aborted ones.
Yes, sir. So actually, then we struggled a lot, and the student also graduated. But recently, I mean, we got that paper.
Okay.
So because I mean,
that
you're
is
from
a very
NRCP, that's why I thought
important
it's directly to talk to you only.
Yeah, that,
Let it be
that's a-
pending. Let it be pending. I will send you the manuscript, the
Ah,
paper, and
that will
then.
be a very interesting thing to see.
So you were talking about miRNA,
Ah,
differentially expressed genes. Now the second question is this, because you checked this pig goat. These are two different species.
Two different species reared in two different geographical zones.
So because these are two different species, so this HSP, I mean, you found something which is common or not?
Common are there.
HS,
Common are there, but variants are also there. Variants also play a very important role. These variants, because there is a situation where one is adjusted to that climate, and the other
Well,
one
so
is a sudden exposure.
why I asked you, because we are having a small piece of work on sheep. So
From different.
yes, from different
Locations?
climatic locations.
Oh, beautiful.
So
Very
this
nice.
part, I don't know. I mean, probably it's not in your mandate or in your area. So
Oh, it is
I
in
was
our
thinking
mandate.
that, I mean,
It is
why can't we connect it?
Oh, it will be interesting. And of course, NBAGR, we have mandate, because
No,
we-
this paper, in this paper you haven't included, you know, the only this sheep and this goat and pig, but you are not talking about sheep. So I was
No.
thinking, I mean, why not to? So
that
this
should be that will be a very interesting thing. That's a transcriptome study.
transcript form publicly available, sir.
Oh,
ऐसे नहीं है sequencing.
okay.
Basically,
You can be in touch. We can
Okay.
discuss further. It will be a very interesting study, because I'm sure this will give a lot of idea about the differential expression, species-wise variation. That's a very interesting thing. Plus, the one information which actually we couldn't assess was that how long it takes for every species to get adapted to that environment. Suppose the generational intervals should be different for each species, is it not? So cattle might be now recently 2 days before we were discussing a similar kind of situation. This Leh Ladakhi cattle is there, and they have HF animals, and we also have the Jersey crosses of this Ladakhi cattle. So they have got a very different genetic characteristics. Plus, there is a saying that these, even though that crosses has happened, HF and not HF pure DFRL, Defense Lab is maintaining this Jersey crosses with the local cattle is available with the public. So how long really takes, because to get that crosses adapted? That will be one question we were trying to find out. We will be taking a study, but they say that the adaptability is not good. And there is a, most importantly, I will tell you, locals and persons who say that the characteristic of the milk itself is different. So yeah, of course, it is definitely true. We have there is a unique characteristic for the milk from that region. That is very much true, and it gives only 1.2 liters, 1.3 very little quantity.
Fat content is high, na?
Fat content is also not very high. The same is case with, that is also a very interesting study. This the metabolome concentration, metabolome and fatty acid profile, they are different. It's a very large sample study. Very large sample study. You can see probably in website, or you can talk with Manishi Mukesh. That is a very interesting study going on, that unique characteristics. The not only the fat and fat protein, these are at superficial level. It's like a, when we go deep, each one of them, which protein, which this thing, so it gives a lot of insights. They say that vitamin concentration is also different. The different B1 is different. The concentration is different. So that gives a altogether a different.
Meenakshi, what name?
Manishi Mukesh. She's a principal scientist there. We can connect you.
I will write
definitely.
to
Definitely. That's a very interesting thing to study, to compare the saying.
different.
Different. Because we have been actually doing the characterization. There is no limit for NBAGR. It is in our domain only. Those work. Pre-registration is there is a policy activity that we continue to do, but we have other things as well. Thank you, sir.
Thank you, sir. Thank you really just telling us about the institute and your research work. So I have like just some very, very small questions. Like
Please.
first, like you mentioned like this non-descriptive mission of NBAGR. So for example, like I really wanted to know like what is the basis where you are just going toward the zero. And the second thing is like this non-descriptive animal mostly generate from like this crossbreeding, like mostly Jersey and HF, and they have uncontrolled, and then finally they ended as a like a non-descriptive. So where like this mission will be like you see on the ground.
Okay, very, very interesting question. The non-descript means there is no description. Okay, a crossbred animal is not a non-descriptive.
But they have like a lot proportion of like
there is a difference between these two. When you cross a Holstein Friesian with a local, so it has got that unique coat characteristics, you know, black and white shades, so that the other one will get. So we can always say that that is a HF crossbred. So that will be written as HF crossbred. That will not be written as a non-descript. Non-descript is the one which you do not have any description. It doesn't have that kind of a characteristic. We say that to have a particular group. It's a what you are talking about is a admixture population. There's admixture is also there, because where a lot of things might be intermingling, and it is very difficult for each one to be told as which one is the dominating one. So based on that, we do characters. We should be able to do to a large extent this non-descript, because otherwise, if you see the census, the ND number is more than like it's a very large number ND. Why ND? Because this has not been included in a different breed. So after we notify it in the Gazette, that goes to the census, and in census that they will be counted separately, those breeds.
Okay, sir. Sir, one more thing, like so you said like you developed like a microarray for the screening of heat tolerant pig breed.
So, that was
so like that's like on transcriptome based.
That is a transcriptome based.
So
yes.
So those like genes are like how you just exclude like the genes which is exclusively expressed like for the heat tolerance only, because most of them you found in like infection and other like stresses like so how you just exclude those things which
those
is just
things. Okay, this microarray was made based on many data. Actually, we had a transcriptome data, we had proteome data, we had multiple data, plus we also had a in vitro as well as in vivo data. So that depends upon these all have been gone into that. So when you take the in vitro data, when you cell culture data, that gives actually a lot of inputs about that many of the things we should be able to exclude what you are talking.
So how many genes it includes?
It is 20.3 K array.
Okay. Sir, one last question. So NBAGR have a lot of like a data, like a cattle data and other data.
Yes.
Still, we are facing the problem to access like a data for indigenous and cattle like or like other animals. So when we will have opportunity to access like
access data.
Yeah.
Yes, we are sitting over a lot of data. Yes, we have a lot of data on different species. Yes, one should be able to do that. We have been analyzing that. You should be. You can collaborate. Why not? You can collaborate with us. We have data, and you can also see that a lot of publications coming each year. Is it not? So that is based on the data. The volume is being processed. The data is being processed on a regular basis. But we should be able to collaborate. Why not? If you wanted to access.
No, for example, like we had like we just make it like publish the genome like and then we have started comparing like to other breeds and making pan genome like similarly if it is like like there's a lot of hurdles come when you go to institute, but when it is publicly available then it's like a more evolved things.
The actual issue come definitely. I agree with you. There are the concern is, I will tell you, the main concern is that people want only data. People, if it is as a collaboration mode, people are welcome and we should be able to do. When you want only data, that becomes a little difficult thing. For example, I tell you, I myself have told them that not to give data or those kind of things where we are not a collaborator in that. Not only this genomic data, people ask for photographs. From that, they wanted to use AI, ML for developing models to identify animals. So everybody will say, "Oh, we will develop a software app where we can identify breeds based on the photograph." Correct? So that much of photographs, हमारे पास जितना है, may not be with other resources. So we have that for all the animals. So 230 breeds means we have for them. So there, if you ask as a collaborator, then it will be easy for one to be given, and there we have a equal say in that. Otherwise, they say that when only आपको photo ही चाहिए, then we say that you don't give. At least now I say that क्या बोलते हैं in no, collaboration is definitely, collaboration is always a welcome thing, and institution grows, each institution grows in that way by collaborating, because everybody has got their own strength, no? That's a really a good thing. If we can collaborate, it's great thing. But only when those kind of things say, this is the one which I always felt that only you ask for the data, बाकी तो हम लोग कर लेंगे.
This is a very important question. So
Sure, ma'am.
we don't have any genomics data.
Hmm.
Neither that data is available in the public. But
Yes, ma'am.
whatever the other data, genomics data, and all the minor data dug by the scientists of this institution, not only during the development of the Indigo chip, but otherwise also, if it can be corroborated with the phenomics data, of course, collaboration is not a
It is
big
sure,
deal.
ma'am.
So that will be a very important
Very
outcome.
important thing, yes.
That's what perhaps Sandeep was meaning, if I'm correct. Right?
Sure, ma'am. That's a, if it is a collaboration, definitely that one should be encouraging that.
Thank you.
And
I just have a
should grow.
small point in your presentation. Thank you for the presentation. In your presentation about NBAGR, you have shown some chicken cell lines. They are in your repository or something. I couldn't understand what is that.
These are germ cells.
Chicken germ
Germ
cells.
cells. Germ cells. We have it with us. Some are with this DPR poultry. They
So that's now I'm clear.
just for conservation.
So now I'm clear. So it's for conservation. So
conservation.
is it available for researchers if they want to do, or what is a policy and strategy of?
I mean, it's actually not a very difficult thing to maintain and develop it. If you
No, that's a different thing. But if you have ready available things, is it
to
ready?
give
To just in a collaborative mode, is there a policy?
No, not really, but we can always do that. In end of the day, I want that NBAGR should do that, because we should be able to give specific, say, Gir cell line or Sahiwal cell line, or this kind of specific breed germplasm. If somebody asks, we should be able to give that. That's the basic end of the day. That is the purpose of the bureau.
So there's one curiosity, like when you say these are the repositories.
Yes, ma'am.
So who are the people who are submitting to this repository?
Okay.
Is it only the ICR fraternity or across the country?
No, throughout the country. Sure. Throughout the country, and our people actually go and collect them from the original source. That is a very sensitive thing, because if we say that it is a gear, means it should be really a gear. And for otherwise, Dr. Sandeep probably was telling about the nondescript or those shades, because Mura buffalo means you have a shade of Mura. So that we have to ensure that really that is Mura. So that's we take it with a pinch of
Thank you very much.
salt.
Yes. Yeah, thank you, Dr.
Thank you
Mohan,
very
for
much.
discussing the important role of climate change in the emergence of zoonotic diseases. So if there are no more questions, let's thank Dr. Mohan for his wonderful speech.
Thank you very much. Thank you for the opportunity.
Thank you. Now
Thank you, CM.
No, no, yes, yes. Yes, yes. Now I request ma'am to felicitate Dr. Mohan.